Hello, welcome to Edexcel Scientific Article 2023 October exam. Here I am going to discuss with you potential questions coming from these two articles. Article 1 it's about your second skin, and then Article 2 discuss about the lymphatic system. Moving forward, what I would like to mention from the beginning, when we take last May June exam series, the research article is about microbiota, gut-brain axis, and the central nervous system. And this was published in a journal called Oncotarget. Now, this time there is a big difference. The difference is your second skin was taken from a magazine called New Scientist. And the second article is taken from a medical journal called Curious, Journal of Medical Science. The title is Meaning of the Solid and Liquid Fascia to Reconsider the Model of Biotech Tensegrity. Now, if you look the relationship between the first article and second article, the first article is about the fascia, and the second article link again with the fascia. It's like a liquid fascia. The solid fascia is the connective tissue. Liquid fascia is the lymphatic system. So that's a connection between first article and second article. And the journal usually address very high-end scientific questions, and the written article is based on a very high level of scientific language. But the magazine is not that. Magazine is in the middle, so it is easily understandable by non-scientific communities such as general public. Now, after having this introduction, I try my best to extract a potential question from a paragraph. Before that, still I would like to give some information about a certain thing. This is the first paragraph. It introduces what is a fascia, kind of a connective tissue. And the main point is it is considered as a sensory organ. So that's the main message. The fascia considered as now sensory organ by the communities, few communities of scientists. When we look the general thing about this article, I would strongly recommend to go through certain level, but certain lessons. But I would like to still discuss with you uh, what is fascia. Now, if you consider this box, gray box, is the 3D structure, and this 3D structure is created by a ground substance. So, molecules, a group of molecules provide this kind of a ground substance, and this ground substance are further decorated by other things like a collagen fibers, for example. So these are the collagen fibers. The collagen are very strong fibers. And next type of fiber is elastin fibers. Now, elastin fiber brings some softness and elasticity to this ground substance. So it's a balance between collagen and elastin fibers. And we have a blood vessels. And nerves. And now we have a cells. So macrophage represent the immune system, fibroblast and myofibroblast and fasciocytes. So these cells are very important to maintain and create the ground substance. Fibroblast, myofibroblast, and fasciocytes. So they can create ground substance, they can synthesize ground substance, they can secrete ground substance, and they can also provide to synthesizing collagen, elastin fibers. And myofibroblasts still bring some kind of contraction and relaxation, represent the muscles. So myofibroblasts represent the muscles, macrophage represent the immune system, fibroblasts and fasciocyte, they are building up and maintaining this fascia. So this is the basic structure of connective tissues, and fascia also contain this kind of basic structure. And we will see in this article why fascia is very important than the other connective tissues. Right now, when we look this tendon, ligament, collagen, elastic fibers like thing, I strongly recommend revise these topics thoroughly: ligament, tendon, collagen, sympathetic and parasympathetic nervous system, inflammation, and skeletal muscle contraction. I'm not going to discuss these topics, as these are the basic things in the syllabus. So you should go again and again and learn the basic theories of these. Right now, coming to the first potential question. Proteoglycans molecule that provide cushioning. The fascia fibers and the soup are both secreted by specialized cells in the tissues, fibroblasts, and the recently discovered fasciocyte. Okay, now certain things are discussed here. Hyaluronic acid, they're called slippery soup hyaluronic acid, and proteoglycans provide cushioning. So hyaluronic acid lubrication, proteinic acid cushioning. For example, we should know why it is important to have this lubrication and cushioning facilities, because fascia uh, covered many structures in our body and our organs, and some of the structures, they are continuously moving, like muscle tissues, they are moving. So lubrication reduce the friction, and cushioning provide the softness and protect the structures. So we should aware of these two words, lubrication and cushioning. So I try to get a potential question. By doing that, I'm also trying to give in this video something not written in the questions, but still you can learn things by watching this video. So that's again different thing. If you get a PDF file with potential questions, typical uh, the pattern is the people try to give you a 30 pages, 50 pages, and probably many questions, like a 50, 60 questions. Now this going to dilute the potential questions, because it include very basic things. Now my target is at least to find more than 60-70% potential question that could come to your exam. But being getting the questions, I'm also highlighting at the same point, you should aware certain terminology and certain potential questions from this video. Okay, now first question coming up. Explain how proteoglycan is produced and secreted by fibroblast and fasciocyte. Why I specially use this question, probably some of them have forgotten you need two theoretical knowledge, or you need one theoretical knowledge. If you forget these things, it is very important that we have to at least discuss here, because one hand you will I can revise your forgotten theory, and the other one is we are preparing for potential question. Explain how proteoglycan is produced and secreted by fibroblasts. Okay, now first you have to understand what is a proteoglycan. I think this is very important that you understand correctly the terminology, because this article is given to you before the questions. That means each terminology you should learn and understand. And even you don't know that proteoglycans is glycan is carbohydrate. Now, molecules which has carbohydrate and protein. So we learn this kind of molecules called glycoproteins. It's the other way, like here is a protein first, glycoproteins, protein comes second. They are different group of molecules, but their basic molecular structure is same. Okay, so we come to the point now. C is a cell. Let's say this is a fibroblast or fasciocyte, and that's a nucleus. So nucleus contains DNA. DNA consists of genes, and the gene is transcribed into messenger RNA. The messenger RNA now undergoing a post-transcriptional modification. The process is called transcription. After that, splicing happened, and the messenger RNA now move into the ribosome. Ribosome is attached to the RER, and messenger RNA now converted into polypeptide chain by the process called translation. And then after that, inside the RER, this is folded to make a tertiary structure, primary, secondary, and tertiary structure of the proteins. And it is packed into the vesicles and moving to the Golgi. So main function of Golgi is protein modification, such as adding glycosylation, carbohydrate, we call glycosylation, add carbohydrate, glycosylation, the glycoprat, and the proteoglycan is now synthesized inside the Golgi as modified by the Golgi. After that, Golgi is again packed this proteoglycan as a final product and then secreted by exocytosis. So I'm not going to write too much here because you can watch again and again the process. Exocytosis is release of molecules in bulk out of the cells. It's active transport process. Okay, so here highlighting the points again, protein synthesized in ribosome, folding take place in RER, Golgi modified the proteins, adding carbohydrate, packed into vesicle, released by exocytosis. Next, fascia, which lines the body cavity and divide it into compartments for different organs. Now, again, one of the function is highlighted by fascia. When it wrap different part of the body, it can compartmentalize. That means it can make rooms inside the body, and these rooms, the wall represented by the fascia. Now, why this is important? Why making compartment important? It's a straightforward question. So describe the importance of compartmentalization of the organ, probably a 1 or 2 marks paragraph. We can say each organ carries out different function, protect delicate internal organs. Organ can expand and contract without distorting other tissues or disrupting the activity of nearby organ. It has small space. This space is very good enough to expand and change the positions like this. For example, take heart. Heart is beating. So when heart is beating, it needs a space and from the lungs. So if the lungs and heart are very closely connected, the heart has again the pressure from the lungs because lung is expanding, heart is also beating. Then there is a problem because both organs at one point touch each other and pressure mount on each other. But if we have a small space between, then the organ can function efficiently. There is no interruptions of the function of the organ, and this is done by making the compartments, and compartments are created by the fascia. So compartmentalization helps to function organ efficiently. So we are now at the 6th paragraph level. The 6th paragraph, it's basically about how this discovery came until 2000. This is not considered as important structure, but some of the scientists, they have done a certain the treatment for patients, and they came up with orthopedic surgeons, anatomists, so these people identified fascia as important place, and they raised these questions. I think most of the information came after 2000. Now suggest why fascia is not studied until 2000. Paragraph 6, 2 marks. Now this is the third question. Why fascia is not considered as a uh, not studied. Now, when we look at Edexcel Unit 2, biodiversity classification, we have a classification system. They have a trend asking why classification system are changing, why new classifications are coming up, why there are two classifications like that. The typical answer is the development of technology. So we can also bring the same context here because it is considered as a connective tissues. Three domain system. When three domain system proposed, the scientific community peer review this one and check whether they can repeat these things, and they approve. So same story here, because it considered as a connective tissues. When they propose this is new as a new role, then they start to peer review and they try to check whether they are repeatable and look scientific publications, other publications, and they come up with the approvals. Because technology was not available to study. Imaging technology and the detection, microscopic like things, not available that time. So as a result, I mean, 23 years ago, 2000, so they haven't understand much about the fascia, and fascia is considered as a connective tissues and it has not much advanced structure. Okay, paragraph 7. Some researchers say that fascia should be considered a new organ, one that is specialized for communication about the body's internal state. Okay, now this is very important point. Scientific community until 2000 considered fascia as a connective tissues. So large group of scientists now thinking this is connective tissue. Now after 2000 studies, scientists came up with this should be considered as a new organ. And when they say this one, they give enough evidence to the scientific community by making this kind of a scientific article, and the community peer review this scientific article, and they check published articles, and they try other groups to repeat this one, whether they can repeat same stories in the other scientific communities, and they come up with the approval. But when it comes to approval here, they didn't give approval yet. They say this is a candidate. Now we have a three group. One is scientists think this is still connective tissue. Scientists now think this is a new organ. Scientists says this is candidate now. So being understand these three buckets, we should prepare for any kind of a questions. So I'm coming up with this question first. So there could be a two questions. So it should like it's a candidate. So it's a candidate, it's a new organ, and other group says, well, it is still same as connective tissues. Now, if you want to make a question for both or all third groups, three groups, explain why scientists are more cautious to define as an organ. 3 marks. Organ is a collection of several tissues and it has a specific function. It is uncertain about the functions such as sense organ, as sense organ has more organized structures. For example, eye. Eye is a very organized structure, retinal layer, and photosensitive cells and their functions like that. Explain why scientists think it's a candidate to name as an organ with 2 marks. Then we can say fascia is a collection of several tissues such as nervous tissue, connective tissue, lymphatic tissues. It has particular functions such as detection of sensation. So as a result, we can consider fascia as a candidate for new sensory organ. Okay, now I stop here, first part of the video, and you go to the second video second and continue from this paragraph 8.